It's Mr. Kohn here with what you need to know about DNA for agri-science or any other class you're in. For DNA, the big thing you need to know is that DNA are the chemical instructions the cell uses to make a protein. A gene is just a section of DNA. Just like a sentence is a portion of a paragraph, a gene is a section of DNA and it codes for a specific protein that has a specific job. A nucleotide is the building block of DNA. A nucleotide is made of a phosphate, a sugar, and a base. The bases are the parts of the DNA that actually code for the information to make a protein. There are 4 bases in DNA: A, T, G, and C: adenine, thymine, guanine, and cytosine. A and T are always found together, and G and C are always found together. A way to remember this is great combinations are always together. Transcription is the process when we make a copy of DNA. A copy of DNA is known as mRNA. RNA is very similar to DNA except for 2 critical differences. Number 1, RNA is single-stranded. Number 2, RNA is going to use a U instead of a T. Instead of thymine, like we have in the blue DNA, we use U for uracil. mRNA is the copy of DNA, and the reason we have to copy DNA is because we only have one set of DNA for every cell. We have to keep that DNA protected from mutations or degradation, and so instead of using the DNA to make a protein, we make the mRNA copy and send that to a ribosome. mRNA, again, is just the copied strand of DNA. When we make a copy strand, we have to put the complementary base for each base in the DNA. So everywhere we see a G, we'll put a C. Everywhere we see a C, we'll put a G. Everywhere we see a T, we'll put an A. And everywhere we see an A, we'll put a U. Helicase is the enzyme that opens and unzips DNA. Here in pink, helicase is seen opening up DNA so that the mRNA can be made. Helicase is sort of like the zipper pull on a pair of jeans. It simply unzips DNA. Polymerase is the protein that actually creates the mRNA. Polymerase, shown here in orange, is what is creating the green mRNA copy strand. So polymerase, in a sense, is just a copy machine for DNA. And the copy that polymerase makes is mRNA. Translation is the process in which we create a protein. The whole point of translation is to make the proteins needed by the cells. Translation always follows transcription. Translation uses the mRNA created in transcription to make the proteins throughout the process. Translation occurs in a ribosome. A ribosome is the protein factory of the cell. In a ribosome, mRNA is read in groups of 3. Each group of 3 is called a codon. For each group of 3 bases on mRNA, an amino acid will be delivered to the ribosome to make a protein. A protein is just a long string of amino acids, sort of like pearls on a necklace. All we have to do to make a protein is deliver the correct amino acids in the correct order. To do that, mRNA is read by a ribosome. For every group of 3 bases, or codon, tRNA or transfer RNA will deliver an amino acid. Every group of 3 bases will be read by the ribosome, and for every group of 3 bases, tRNA will deliver an amino acid until we have a string of amino acids that makes a protein. If we look at the whole process together, we can see transcription occurring in the nucleus and translation occurring in the ribosomes. The first step is to open DNA using helicase, make our mRNA copy using polymerase, and then send that mRNA copy outside of the nucleus. The mRNA will leave the nucleus and go to a ribosome and enter the ribosome. The ribosome will read the mRNA in groups of 3, and for every group of 3, our orange tRNA will deliver a pink amino acid. Over time, those amino acids will form a chain, and the chain of amino acids are our proteins. Transcription occurs in the nucleus. Translation occurs in the ribosome in the cytosol. mRNA and DNA are a part of transcription. mRNA, the ribosome, and tRNA are a part of translation.